אפשר להתחיל? ערוכים? אנחנו מתחילים. נראה לי שאנחנו יכולים להתחיל בעברית, נכון? או שאולי הם ירצו לראות את הוידאו, אז אוקיי. טוב. Okay. I remind you, this is the second talk about the olfactory system. And we're going to have, I'm going to talk about the main olfactory system now. On Wednesday, Yoram Ben Shaul will give you the vimoronasal system. And we're going to have paper session next week and finalize this and move on to auditory, which is the last one. Last lesson, basically we talked about odors and we talked about olfactory receptors and olfactory receptor neurons in the epithelium. And we found out that each olfactory receptor neuron expresses a single out of 1,000 receptors, and but each neuron can respond to many odorants, and different odorants are recognized by different combinations of receptors, and indicating that the olfactory system uses this combinatorial code. Okay, so this is the first, really the first step of transformation from the chemicals to the nose, to the epithelium. And now we want to step into the nervous system, and we're going to do it slowly and hand in hand with perception, as we actually already have seen with the mutations in the human perception. So most of what we know in olfaction is solved in different types of animals. In some animals, the situation is rather simple, or we think it's simple. Actually, I had a third slide which shows you that it's not as simple, but if you're interested, you can look into the paper. So mostly we understand the olfaction in mammalian systems, and Drosophila perhaps is the best. In many cases, Drosophila and C. elegans are the ones who are paving the road into how we study these senses. In the worm, it's a little bit different. As you know, it has only 302 neurons, and now there are some neurons which we already know are responsible for specific behaviors. So, for example, I don't know if you've ever seen a worm lab. No? So there are many, many students, many postdocs who are screening. So they have a lot of mutations, and they're screening for a phenotype. And sometimes they spot a specific interesting phenotype. This can be a phenotype in the circuitry. This can be a phenotype in the behavior of the animal. And once they find it, then they start going after the mutation and where the problem is. And normally, they see things like this. So, for example, we now know that out of the 302 neurons, there are specific neurons, olfactory neurons, that are responsible for very important behaviors, even in the worm, or mainly in the worm. It's not a very interesting animal in terms of its behavior, but attraction and repulsion. So we know that a neuron called AWA is responsible for attraction, AWB for repulsion, etc. And how this is done, you can easily make out knockout worms. So, for example, this is a bioassay, a behavioral test, where an animal is put here in this small dish, and is filmed. And you can put, this is the control side. You can put an odor on the other side and score whether the worm will find itself to be attracted to the odorant, be repulsed by the odorant, or all over the place. And as you can see, if you take a wild-type worm and put it in the center of the field, most of these worms, most of the time, will be attracted to an odorant called diacetyl, okay, which is some odor that worms like very much. And if you screen and you find mutants that do not respond to this odorant, you find out that, for example, the knockout is in some odorant receptor. And if you look at the knockout of of this neuron, okay? This is you can knock out a specific receptor from the AWA neuron, okay? So in the wild type, this odorant receptor number 10 is expressed in AWA, but not in AWB. So you can find a mutant that has no odorant receptor number 10 in this example, and you look at the expression in AWA, there's no expression, and you put you put the worm into this small field, and you see that it will spend time on either side equally well. And the beauty of these types of animals and experiments is that you can now do gain of function things. So basically, you can take this receptor and exogenously express it in a different neuron, okay, which is known to be a repulsion neuron in this example. And you put them in the arena here, and you see that now it is repulsed by the odorant. Okay? This means for us that it is the downstream neural circuitry that is responsible for the behavior in this case, okay? Because it doesn't matter what odorant is expressed, it matters which neuron was activated. So if neuron AWB was expressed, then now the worm was repulsed, and if under normal condition AWA is expressed, then it's attracted. And again, in these types of models, the experiments are very, very robust and strong and convincing. You can see many of them, therefore, in journals like Nature, because it's very clear-cut. You see the behavior is very clear-cut. Variance is very small. This is attracting. This is repulsing. You take the receptor out. You knock it out. You introduce it back into AWA, into these animals, and you can get full recovery of the behavior, etc. Okay? Yeah. So that's the beauty of these systems. Each neuron can be identified and can be accessed. So out of the 302 neurons that the worm has, they have genetic access to each and every neuron. And I can play genetically with anything I want. I can express into this neuron, and particularly to that neuron, based on genetic profiling of the single cell. And in many cases, it's not true that you can have full access to all the neurons, but in C. elegans, most of the neurons, in Drosophila, a lot of the neurons. You can play with the timing, for example, so you can express at a given time a transcript, whatever transcript you want. And because these neurons are born out, let's say out of the 302, they are born consecutively, and you know exactly when you can activate the transcription, you can get activation in specific subsets of cells. But these systems, it goes from yeast, shmarim, which is like the best genetic model. You can do whatever you want. And we go up. C. elegans is the one with the nervous system that we think we can understand. But many people say, this kind of animal cannot really, is not a very good model for the brain, on one hand. On the other hand, if you believe that there are certain principles in nervous systems, you may find them here as well. But in terms of experimental access, there is no competition. Okay.
There's no variability between individuals? There's no plasticity?
In these animals, not much plasticity. You can teach. Eric Kandel made a big career with the mollusks that can learn, habituation, adaptation. But it's not, if you look at plasticity and learning, these animals are not very sophisticated. For example, I'll give you an example. One thing in developmental neuroscience is that if you look at mammalian systems, they are extremely influenced how the nervous system is wired up by the experience of the animal. Okay? If you take a baby, you put him in the dark for 10 years, he will be retarded by, there's no question about it. Okay? Experience is an extremely important part of the development. In flies and worms, not so much. If it was raised in an enriched environment or not enriched environment, etc., these systems are much more hardwired. Therefore, one can find identified neurons at the level of individual cells and they have their great advantages. Things like development, evolution, even things like a synaptic release. And now even in neural circuits, okay, you'll see as we go along in olfaction as well that these are systems that teach us a lot about how the nervous system computes information. And there is some variance, but very little. So I think if you take worms, 99% of them will have 302 or 301 neurons, something like that. Okay. But what we know more about, and for example, the worm, they don't have action potentials. They're neurons, okay? They have only graded calcium responses, which makes people who think that action potentials are important a little scratchy. So I think the closer look to a more sophisticated nervous system is that of Drosophila. They have certainly many, many more neurons in the millions. Again, not like the mouse. And certainly it's hard to put this in close approximation to human, but I'm having a hard time to put this in close approximation to human. So anyway. Okay, so we know a lot about these two nervous systems, the fly and the mouse. Actually, Rotem will talk about a different species, which is the fish, and each one has its own differences that may be important. For example, in computational neuroscience, we know a lot from a researcher called Gilles Laurent, who was in Caltech and is now in Germany. And all the foundations of his concepts were based on the locust, which has its advantages experimentally, but in the olfactory system, but also has very important differences. Okay, but these two systems of the fly and the mouse are very much alike. The mouse, although it has 1,000 receptors or 1,200, the fly only 60 olfactory receptors, but he also has a one olfactory receptor, one neuron rule, for example. Also, his axons from a single olfactory receptor converge into single glomeruli, and that is an important anatomical or circuit-based evidence that is very similar to the mouse. So we know quite a bit about this and making the jump, although they are not similar in all levels. We will see a few as we go along. Here too, like the advantage of the fly is also we have genetic access and sometimes very robust behaviors. You can see this. This is just a 2007 paper, but there are now, every year something new is coming out with a single odorant receptor that is responsible for the full-blown behavior of the animal. In this case, it's a receptor called GR21, and you can put channel rhodopsin in it and make some behavioral apparatus for the fly to go right or go left, again, attraction or repulsion. And you can, this one is avoidance, okay? So we know that flies don't like CO2, they will avoid CO2, and here they show very robustly that a given olfactory receptor population is responsible for the avoidance behavior. Okay, this is just a few controls. Here is the CO2 in a fly that has channel rhodopsin in these olfactory receptor neurons, a single ORN. Here with the CO2, here only with light. So you see again how robust the behavior is when you look at animals like the fly. So again, in a similar concept as the worm we saw before, very clear, very robust genetic access and behavior. In Drosophila, they started to map out the olfactory world of the receptors. This is one of the first or more comprehensive screens of odors. In this case, they tested 100 different smells, odorants, while they were patching specific olfactory receptor neurons on the nose of the fly. By the way, the nose of the fly is on its antenna in this case. Okay, so the receptor neurons, olfactory receptor neurons, are on the antenna. And here they screened 24 out of the 60 olfactory receptors. Here too, Again, you cannot do something very easily like that in mammals. Here you can take a fly, you can cut his head, put in a microelectrode exactly on the same position on the antenna, and you can target a specific olfactory receptor. So they know that, all these recordings were made from the 2A neuron, and the 7A neuron, and the 9A neuron, etc. And this way they mapped the responses of each of these 24 receptors to 100 odorants. Okay, again, this goes to some discussion we had last time about the size of the olfactory world, but the basic finding here is that the yes. Yeah, so this is alcohols and aldehydes and acids.
No, on the table itself, like
Here.
what's the difference between 1 plus and
This is the magnitude of the spiking response of the neuron. Okay, so yeah, the number of spikes, well, it doesn't really matter. Okay, in this case, let's say neuron number 7A responded very strongly to this, etc. So you can see some neurons have broad receptive fields, some are very specific, very dedicated channels of activity, and therefore the accepted model, if you will, is that if you look, let's say, at 2 axes in the olfactory space, some neurons will have broad receptive fields, wide receptive fields, some olfactory receptors. Some will have very tiny and will be very dedicated to a specific odorant. You will see next Wednesday in the vimoronasal system that most of the cases in the vimoronasal system is like that they have very fine responses to specific odorants, and they're responsible for a full, in their case, sometimes a full behavioral response when they are activated. But some neurons have wide receptive fields, some have narrow receptive fields. And the idea here, although it's not completely proven, is that the animals, and this is an evolutionary thing, will have more information in a given axis based if you're a bird or a fly or a mammal or a fish, of course, okay? Because the repertoire of responses to odorants in a fish will be different than for us because we're terrestrial animals, etc. Okay, but generally speaking, now you look at the olfactory system. It's not that each one has the same size of receptive field. It's a very complex sheath of neurons. Some have very wide receptive fields, some have very narrow receptive fields in the system. Again, this is not fully proven, certainly not in mammals, because you saw we're pretty much stuck to the Petri dish with expressing in heterologous systems. In Drosophila, this is based on this screen. Actually, in mice, there's been in 2011 another screen, actually by Linda Buck, who got the Nobel Prize from her own lab. And they tested 3,000 olfactory sensory neurons to 124 odors, although I think they did this randomly, these 3,000. And they also support this kind of model in mammals. So it seems that this is generally true for the olfactory system. Okay, the more robust experiments came, like I said, from Drosophila. And the first person who actually, so let me back up a little bit. Drosophila was great because you can do genetic manipulations, trace the neurons, the anatomy, blah, blah, blah. But there was one big caveat in Drosophila where all of us in the mammalian systems always dismissed the Drosophila system, is that they could not record from the neurons in the Drosophila. And the big breakthrough came from a postdoctoral fellow in Caltech, in Gilles Laurent's lab, called Rachel Wilson. And she decided that she's going to record from Drosophila neurons once and for all. And it's a problem because in Drosophila, the cuticle is on the outside. So the bones are on the outside, and inside is the... So you crack the nervous system, the brain, and there's all kinds of issues of osmolarity and pressures. You couldn't really record. She was smart enough to solve it, and she started recording from Drosophila, and that was a big breakthrough in the field of Drosophila in general, because they can now enter into the area of computational neuroscience and what computations are carried on by these neurons, etc. Okay, until then they were pretty much stuck maximum with calcium imaging, which has its limitations. And electrophysiology is the gold standard. So for example, let's see some of the experiments that, this is now from her own laboratory. Rachel is now in Harvard. She is a very successful researcher to this date, in this case in her first year starting to work on the olfactory system in Drosophila. So this is the schematic of the nervous system of Drosophila. On the top are the olfactory receptor neurons. You remember that each one sends axon to a single glomerulus, and there are many of these, many orange ones, many purple ones, etc. And these are the output neurons from the olfactory bulb. In this case, the antenna lobe, which are the homologous structures. So what she can do, and what we are having very hard time to do, is now not only does she know all the repertoire of odorants from the Carlson paper, from this, okay, now let's say she targets figure 43A, she has at least for 100 odors, she knows the receptor field of these cells, so she can choose. But moreover, because the nervous system is so simple, she can patch and record from the presynaptic neuron and the postsynaptic neuron of the same channel. Okay? So she can go to olfactory receptor, this green one, and record from its cognate mitral cell. Okay? Its partner, the one who is sending the dendrites into a single glomerulus. I'm not sure if I said it last time. Each mitral cell sends a single dendrite into a single glomerulus. Okay? I think I said it. If not, this is an important thing. Olfactory receptors in the epithelium will send their axons converging into a single glomerulus. Okay? Glomerulus is just a structure of synaptic connectivity. There are no cell bodies in this structure. It's ball-like structure. And the output neurons from the mitral cell send a single big dendritic tuft here. They also have lateral dendrites, which we will talk about soon, but this goes to cortex. So basically, this forms a functional unit in the olfactory system, okay? A single odorant receptor and a single mitral cell. What she can do, what Rachel Wilson can do in Drosophila is actually record from the input and the output channel simultaneously. Okay, with very high accuracy, because she knows exactly that this mitral cell goes here, etc. And this is what she finds. When she records from olfactory receptor neurons, this is the, there are about 3 projection neurons, 3 mitral cells, or in Drosophila they're called projection neurons that send their dendrites into a single glomerulus. This is the responses more common to the olfactory receptor neurons. And this is what one would get if he records, or she records, from the projection neuron, from the postsynaptic cell. Yeah.
The local neuron and the figure is usually inhibitory one?
We'll talk about that, okay? Because this is exactly what we're looking for. First, before you start to go into the system, into the circuit, one of the most important things, generally, when you go into a neural circuit and want to understand what's happening there, is to record the input-output function. What is the input? What is the output? And therefore, you can start thinking of what is the computation that is going on there. And there, of course, not a single computation. There are many computations. It can be modulated. It can be dependent on behavior, etc. But this is the first example in the olfactory system where we recorded an input and a cognate output channel together. Okay? We know there are interneurons. We'll talk about the interneurons shortly. But that's the next level. Let's first look at the transformation. Yeah.
Can you repeat the diagram, please?
This diagram? So the olfactory receptor neurons, they sit in the epithelium here. Each one expresses a single ORN. Were you here last time?
Yes.
Okay. And all the neurons from the same, that express the same ORN, send their axons into these glomeruli. Maybe I'll just give you an image how it looks. It's Let's go 2 slides up. This is a beautiful image that one of my friends took in the postdoc. This is a mouse now, and you see all the olfactory receptor axons. There are about 15,000 of them in the green. They're coming from the epithelium, which is somewhere where they build